All right, and then I would like to announce Johanna Okerlund from Wellesley College, who will be presenting Eugenie.
Thank you. My name is Johanna Okerlund. I'm from the HCI lab at Wellesley College, and I'm here to present a system, Eugenie, a system for biodesign that we've been working on for the past 2 years. So in this presentation, I will first start with an introduction to the biodesign process. Now, I know what some of you are thinking, and if you're afraid of the word biodesign, don't worry, I promise it will be very brief and high level. And if you came here really excited about biodesign, then I apologize that this will be so brief and high level. But then I will present Eugenie, the first iteration of our system, implemented as a multi-touch application. And then I will present Eugenie++, a tangible interaction system for biodesign. So the biodesign process begins with a goal, to create a biological system that has a particular function. For example, biosensors, which are organisms that can respond to different environmental cues. For example, maybe some bacteria that turns a different color in the presence of a toxin. So biologists can make some pretty crazy cool stuff. To achieve these goals, the biologist works with something called biological parts. Parts are sequences of DNA that are organized based on their function. Don't worry what the functions are, just think of these parts as building blocks, biobricks, or even Legos, if you will. So these are the 4 generic types of biological parts: the promoter, ribosomal binding site, coding sequence, and terminator. And of course, there are more specific instances of each one. So the biologist arranges these parts to form a biological device. The parts in a device have different properties, such as neighborings, orderings, and orientations. But not all orderings of the parts constitute legal devices. So that's one thing the biologist has to be careful of. And of course, the devices get much more complicated as the biologist adds more and more parts, and also relationships between parts, shown here by these lines. So there is an existing language for biodesign. This is not something we developed. This is Eugene. Our prototype is Eugenie. So Eugene is a text-based language for biodesign, where the biologists specify which parts they would like to use, and also which rules they would like those parts to follow. The system then generates all of the biological devices that follow those rules. The biologist looks at these devices and adds more rules as necessary until they have the devices they want to work with. So our lab at Wellesley College has had a design partnership with synthetic biologists at Boston University for the past 3 years. And we've spent a fair amount of time in their lab, studying the work that they do, learning about their processes and their research. We've also spent time together designing software and actually deploying it and studying it in their lab. In a recent workshop, we discussed the biodesign process, and together we came up with a number of goals that a system that would support biodesign would have. One goal is integrated workflow, that the biologists use information from all over the place. There's the parts database, and then there's all sorts of information online, and we would like all of that to be in one place. We would also like to support the rule-based design of Eugene, since that's the workflow the biologists are used to working with. We would like our system to foster collaboration. Right now, what our biologists do is that they gather around a whiteboard and sort of think of a general idea, and then one person goes off by themselves and codes it. We would like that entire process to be a little bit more collaborative. We would also like our system to support constructivist learning. That our biologist friends, a lot of them are undergraduates there just for a summer program, and they're very new to all of this stuff. So we would like our system to not only be a tool in the lab, but to be a learning tool as well so they can learn about the biology. And lastly, our biologists required that whatever we implemented would be able to be supported by multiple platforms. So the first iteration of our system, we used a 40-inch multi-touch surface, the Microsoft Surf 40. And the workspace looked like this. You can see that there are 5 different sections, which are separated by sliding panels, so the user can view either one or multiple things at once. On the left is the toolbox, where the user can search the parts and choose the ones they would like to work with. They then define the structure rules of the device, as well as the behavior. And as they're doing all of this, the corresponding Eugene code is generated and also highlighted. The user can also view the results in the results panel, all of the corresponding devices that are generated by those rules. So for the evaluation of the system, we had 15 biology undergraduates. 12 of them worked in pairs, and 3 were a little more experienced, so we had them work alone. We gave them all an introduction to the biodesign process, similar to what I just gave you. We then gave them a walkthrough of how to use the system. The task was then for them to build this biological device using the system, something that synthetic biologists generally do. All pairs successfully completed the task. Pairs spent 42 minutes on average, and individuals spent 41. So I have here now a video of some of the interactions with the system, and this video also presents the results of our evaluation. So these are the sliding panels as the user manipulates, drags apart, specifies the structure, adds more detailed information in this corresponding part sheet, defines the behavior relationships between the parts, views the generated code, and also studies the results. So some points of improvement. One was inclusion. We had a multi-touch application with small target sizes. Of course, occlusion was a problem. Another was a lack of constraints. That it's difficult to know what legal actions are and what can be performed, what makes sense meaningfully. Another problem was aggregation, that there's no way to combine complex things into something simpler to manipulate. And also persistent representation, that a lot of information was actually represented in these part sheet and not on the part itself. So to summarize, there were a number of weaknesses of our system: occlusion, persistent representation, lack of constraints, and aggregation. We could have iterated upon this design and improved some of these features, but instead we decided upon an alternate approach, which was to offload some of the interaction from the surface to the area above and around the surface. And we did this with tangible active tokens. So this is the second iteration of our design, Eugenie++. You see here the tangible active tokens, which are microcomputers that have sensing capabilities such as shaking, tilting, and touching. We chose to use tangible active tokens because biological parts lend themselves well to the metaphor of building blocks. And we like the idea of the user being able to directly manipulate the parts they are using and physically build these rules. So the way the system works is that the user physically builds a rule and then stamps it onto the surface. The surface then interprets what the user just did and displays the corresponding Eugene rule in text formation. The surface also has a view where the user can view the corresponding results, all of the devices generated by these rules. So because we offloaded so much of the interaction to the cubes themselves, we needed a way for the user to be able to search lots and lots of parts on the cubes. And we decided on tilt to search. So the user first tilts to search between the 4 generic parts. But there's also a database of 3,000 or so parts that the user might want to search between. And that would be a lot just to tilt through. Luckily, this dataset is hierarchical. So for each generic part, there are types of that part. For example, yeast-driven promoters. Then for every type of promoter, there are specific instances of that. So to search something below the generic level, the user neighbors a cube under the first cube. Then that second cube is used to traverse the next level. And the user does this until they have the desired part. And this is a video of some of these interactions. Browsing the generic parts. Now searching the kinds of that part. And finally, searching the specific parts. Users can also shake to clear the cube at any point and have it mean something else. So the structure rules of our system were designed with a combination of active and passive tokens. Our design principle of these was to have the physical constraints of the tokens match the syntax rules. So for example, starts and ends with are just pieces on the end of each piece so that nothing else can go there, so it makes sense. Also, to indicate that a piece is reversed, the user just reverses the token and stamps that to the surface. For the behavior rules, we decided to use gestures performed on the surface with the pieces in question. Represses and induces are relationships that indicate either more or less output. So we decided on clockwise or counterclockwise motions, either indicating turning up or turning down the output. And here's a video of some of these rules being built. So if the device does not contain a part, you put this not tag on it. If you want to specify that a device has a certain number of a part, you can use this equal sign. And then a number dial to indicate how many. And if a design starts with a part, you use the starts with tag. So you build the rules and then you stamp them to the surface. So for our evaluation of the system, we had 18 users, all of whom were science students, but who had different levels of experience with biology. So we paired people according to experience. For the first task, we wanted to see what users would do with the tokens without having been given any instruction. And we used a logical test to do this, so they wouldn't have to understand synthetic biology. We had them build these logical statements using the pieces shown. The second task was closely related to the task we used for the first iteration of Eugenie. We gave users an introduction to biodesign. We gave them a walkthrough of how to use the system. And then the task was for them to build these biological devices with the system. Pairs spent 36.5 minutes on the tasks and all successfully completed both. Users found the tilt feature very easy. They figured that out very quickly, and also figured out the logical operators. But they had difficulty with a few of the other operations, which I'll talk about a bit later. So our results indicated that users used the physical tokens to think. A lot of groups would sort them based on size or function. They would also hold or point to a piece while they were considering it, or while they were talking to their partner. We also saw groups making gestures for behaviors. That one group, while they were considering if they should do a repress, they would actually make the repress motion in conversation. There are also several limitations of the system. One was the limited interaction space. The bezel was only 4 inches, which really limited the amount the user could explore and sort the pieces. They really were limited to sort of horizontal interaction. We also expected the users to manipulate the piece a lot in air, but they didn't do that so much. They would leave them flat on the surface, and we suspect that's because of the flat, puzzle-like form factor of the pieces. Another limitation was rule persistence. That in our system, when the user builds a rule and stamps it to the surface, it's permanent, and the user can disassemble the rule and use the parts for something else. Except we noticed that people would stamp the rule and then leave the parts still assembled. And perhaps that was because the link between the digital and physical representations was too abstract, and they wanted to have the physical reminder of the rule. Another limitation was user expectations. That for the logical test, we noticed users putting the not block next to the thing it was negating, because that's where not goes in a sentence. Even though if you look at the block shape shows that the not should be connected to the top. Users also had trouble with the if-then. Most figured out that if-then was indicated by this arrow, but all groups were also looking for an if block. Because the sentence had an if, they wanted the rule to have an if as well. All right, so stepping back from the individual details of our implementation, Eugenie is a case study in the application of tangible interaction with active tokens to the realm of database exploration and the formation of complex queries. So by evaluating Eugenie, it highlights some of the challenges and opportunities of applying this approach to this realm. One of the challenges is rich representations, that there's a lot of information that needs to be represented in a meaningful way. In our system, we first had to give the biologist the way to search all of the parts. We then had to represent the parts. And luckily we could do this visually. But there was also a lot of information associated with each part, not all of which has inherent representations. And lastly, of course, we needed to give the user the tools to connect the parts, to make rules and have meaningful interactions. Another challenge is closing the gap between tangible and digital representations. That in our system, the physical representation was the Eugenie rule, and the digital representation was the corresponding Eugene rule. But this connection was too abstract for the users, and they needed something to bridge that gap, maybe something visual on the surface or something like that. Another challenge is the aggregation of complex configurations into simpler representations. That if the user builds something long and complicated and wants to refer to that thing as a whole, there should be a way for them to sort of condense it down into one token that they can then manipulate. We also discussed the opportunities of applying tangible interaction with active tokens to the realm of database exploration and the formation of complex queries. One of the opportunities is scalability. That with dynamic binding, we can represent a large number of things with just a small number of tokens. In our case, even 3,000 maybe. Also with dynamic binding, if users want to, in the course of interaction, change the meaning of a piece, they can. Another opportunity is to explore a new interaction space, the space around and above the surface. We noticed users using this space to explore and sort, and further studies could be conducted to better understand those interactions. Also, new systems could be developed that actually leverage those interactions in that space. There is also the opportunity to enforce syntax with physical constraints. to make interactions even more meaningful. There's also the opportunity to explore how to provide tools for users to think through action, giving them the means to think externally. And of course, supporting collaboration, that our system has multiple points of entry, that multiple people can be working around the table, exploring a database at the same time. They can either work on the same rule at the same time, or work separately and then collaborate what they have found. So in conclusion, we implemented, developed, and evaluated multiple iterations of a system for biodesign. And our work Eugenie, our system Eugenie, is a case study in the exploration and the use of tangible interaction with active tokens for the exploration of large data sets and formation of complex queries. Thank you for listening, and the floor is now open for questions.
Thank you. And there's one question.
Hi, Roll Vertigo, Queen's University. Have you heard of Symbiota?
Sorry?
Have you heard of Symbiota?
No.
Yeah, it's a company that does like a platform for essentially programming DNA as if it was an object-oriented code base with a web-based interface. And then you can send it off, and they can even synthesize the DNA if you want to. So you could do subroutines and things like that. But I did a workshop with them a couple of years ago, and one of the things that struck me is that it's one thing to know sort of what it is you want to program to begin with, which is kind of what you instruct, right? But it's quite another to actually make it happen. So in this workshop, there was like magnetic markers that were like inserted to sort of like have strings of DNA together in a test tube. You had to shake it, and then you had to wash it out. You had to sometimes repeat these kinds of steps. To what extent have you considered teaching people those kinds of skills? Because if you have tangibles are perfect for that, and one of the issues is that there is limited shelf life, these components may be expensive, and you may want to try or at least get some of these skills before you do your first real session. Have you thought about this?
Directly, no, but we have sort of in a similar vein to that, but sort of a lower level, sort of interactive tokens and things that can teach about the biodesign process in general. So we have, active tokens where the user can sort of put a gene into a bacteria and then pour that into a dish and then see the interactions. So yes, at a basic level, we have thought about that, but not sort of the more iterative and more complicated processes that more advanced biologists would have. That's cool, though.
Yeah, it might be nice follow-up, because I found that very challenging. Is this just to, I mean, the yield is like 20% or something like that.
Yeah. Thanks. Maybe I can ask you a question. So it seems from your work that you mainly targeted like students who are learning about this field. I was wondering if you had some ideas of if like the more senior people in this field could also benefit from the tangible approach.
That's a good question. A lot of times taking tangibles into other fields, they're a little resistant to it. They're so used to working with the text-based Eugene, they're like, no, we don't want tangibles. But I think there is value to it. I think that there's a huge value of a whole bunch of people working on something together. So I think it would take a little bit of getting them used to the idea, but I think that it would definitely be valuable for any community, yeah.
Next steps.
Yeah.
All right. That doesn't look like a question. Okay, then thank you very much.